Hello friends, I am Dr. Sanjala Babuta from Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdard. Today we will be discussing about the module Physico Chemical Factors affecting drug absorption. In this module, we shall be covering and understanding the various physico-chemical factors which affects the absorption of the drug, like the drug solubility and the dissolution rate, the polymorphic crystals, hydrates, solvates, the chirality of drug, the salt form of the drug, the pKa of the drug and the pH partition hypothesis, the stability of the drug in the gastrointestinal tract, the surface activity, complexation, absorption of the drug. There are a number of factors which affects the absorption of a drug from a dosage form or from the drug itself. The first are the physico-chemical factors. The various physico-chemical properties of a solid drug particle, like its solubility and dissolution rate, its particle size, its form, that is whether it exists in the crystalline or the amorphous form, the polymorphism, pKa, its lipophilicity, pH, stability, all play a very important role in determining the absorption of the drug. Thus, these factors are important to consider in the design of a dosage form. The first factor is the solubility of the drug. Let us understand what solubility is. Orally administered dosage form must dissolve in the gastrointestinal fluid before its absorption for systemic activity. As a rule of thumb, drugs with a water solubility of more than 1% are not expected to present dissolution associated absorption problems. But before applying this rule, one must consider the drug dose and its stability in the GI medium. Highly insoluble drugs require large dose to show good absorption, whereas drug exhibiting instability in acidic environment of stomach, high solubility and fast dissolution show reduced bioavailability. Because of these reasons, aqueous solubility is a beneficial biopharmaceutical parameter. A basic drug is more soluble in an acidic medium, forming a soluble salt. On the other hand, an acidic drug is more soluble in the basic conditions like intestine, forming a soluble salt. The solubility pH profile provides an idea about the completeness of dissolution for a drug dose in the stomach or in the intestine. Solubility of a drug can be enhanced by the addition of an acidic or basic excipient. For example, solubilization of aspirin may be enhanced by the addition of an alkaline buffer. In case of designing controlled release drugs, the buffering agents may be used to modify or slow the release of fast dissolving drugs. Dissolution is defined as the process of solubilizing a solid substance in a solvent. It involves transfer from the surface of the solid to the solvent. In case of dosage forms which are administered orally, the following determines the rate of absorption: dissolution rate and permeation rate through the membrane. The following figures illustrates the various steps which are involved in the absorption process of the drug after oral administration of a solid dosage form. There are various theories which are related to the dissolution of the drug, and the three important theories are the diffusion layer theory, the DANCKWERT'S theory and the interfacial barrier theory. So let us understand in detail about the diffusion layer theory. This theory consists of two steps as shown in the figure 2. In the first step, there is formation of a diffusion or stagnant layer when the solid drug comes in contact with the GI medium. The diffusion layer forms at the solid-liquid interface. This step is generally rapid. The next step is that from the diffusion layer, the soluble solute diffuses to the bulk of the solution. This step is usually slow. In case of diffusion controlled dissolution, Noyes-Whitney equation explains the dissolution rate. The Noyes-Whitney equation is coming on your screen. In this equation, dC by dt is equal to drug dissolution rate, D is the drug diffusion coefficient, K, W by O is the partition coefficient of the drug, Cs and Cb is the drug content in the stagnant layer and drug content in the bulk of the solution at the time t respectively. Cs minus Cb is also known as the concentration gradient. A is the surface area of the drug, V is the dissolution media volume and H is the stagnant layer thickness. All these parameters play a very important role on the drug dissolution, and the effect of these different parameters on drug dissolution is explained in table 1. The first parameter is the diffusion coefficient, which is represented as D. Higher the value of D, there is a rapid dissolution rate of the drug. The second parameter is the viscosity of the dissolution medium. As the viscosity of the dissolution medium increases, there is a decrease in the diffusion of the drug from the stagnant layer to the bulk solution. The third is the surface area of the dissolving solid. More the surface area, rapid is the dissolution. Surface area of the solid can be enhanced by micronization of the drug. The next is the water-oil partition coefficient of the drug. More the value of the partition coefficient, higher the hydrophilicity of the drug, thus rapid is the dissolution. The next is the concentration gradient. Passive diffusion is the mechanism of drug transport where the drug diffuses across the biological membranes in a direction of its concentration gradient. Higher the concentration gradient, faster the drug diffusion and dissolution. The concentration gradient can be increased by enhancing the solubility of the drug and increasing the dissolution media volume. The next factor is the stagnant layer thickness, represented as H. More the thickness of the stagnant layer, lesser the drug diffusion and dissolution. Increase in the agitation of the dissolution media leads to a decrease in the value of H. The various factors which affects the dissolution of a drug are the particle size and the effective surface area of the drug. Particle size may affect the solubility of the drug and thus the dissolution rate and absorption. According to the Noyes-Whitney equation, more the surface area, faster the dissolution rate. As the surface area increases, by reducing the size of the particle, faster dissolution rate is achieved. More effective surface area causes greater intimate contact between the solvent and the solid surface, thus more dissolution occurs. But it is only in the case where the particle size gets reduced below 0.1 microns that there is an enhancement in the intrinsic solubility and drug dissolution rate. The surface of such small particles exhibit energy more than the bulk of the solid, leading to more interaction with the media. For example, micronization of poorly soluble aqueous drugs like chloramphenicol and glycopholin leads to superior dissolution as compared to simple milled form of such drugs. Sometimes it has been reported that micronization results in decrease in dissolution rate. The reason for this includes extreme reduction of particle size imparts charge to the surface which prevents wetting. The hydrophobic surface of the drug absorbs air onto their surface, thus leading to inhibition of wettability. Drug particles reaggregate and form a larger particle because of their high surface free energy. The absolute surface area of lipophilic drugs can be changed to effective surface area through the following strategies: by using surfactants which decrease the interfacial tension. For example, Tween 80 enhances the bioavailability of phenacetin by improving its wettability. By using hydrophilic diluents because they make a hydrophilic coating over the lipophilic substances. Examples: by using diluents like PVP, which is polyvinyl pyrrolidine, polyethylene glycol, dextrose, etc. Partition coefficient. Biological membranes are usually lipoidal in nature. Therefore, lipid solubility of a drug is a major parameter for its absorption. Consider example of opium alkaloids like morphine, thebaine and codeine that comprises of 2, 0 and 1 hydroxyl group respectively. It is reported that the absorption rate decreased by enhancing the polarity of the molecule. Moreover, lipid soluble drugs like quaternary ammonium compounds, aminoglycoside antibiotics, filtered at glomerulus, back diffuse into the tubules as 99% of the filtrate is not reabsorbed. On the other hand, non-lipid soluble drugs and highly ionized drugs are not able to get reabsorbed. Therefore, excretion rate of lipid soluble drugs parallels glomerular filtration rate. Alteration in urinary pH influences the tubular reabsorption of drugs that are partially ionized. Weak acids ionize more and exhibit minimum reabsorption in alkaline urine. Weak bases ionize more and exhibit minimum reabsorption in acidic urine. The lipids that are present in living membranes are difficult to obtain in pure, for that's why one can determine the relative lipid solubility of a drug by checking its distribution between the immiscible organic solvent and water. When a drug is added in two immiscible phases that are in contact with each other, the solute will distribute itself between these two phases in a fixed ratio. This ratio is called as the partition coefficient. Partition coefficient is independent of the amount of dilute solutions of a given solute species. The following organic solvents can be utilized in the evaluation of partition coefficient, like N-octanol, chloroform, carbon tetrachloride, amyl acetate, ether and isopropyl myristorate. The benefit of partition coefficient data is to determine the absorption potential of drugs. For example, barbital exhibit partition coefficient, that is a oil-water coefficient of 0.7 and an colonic absorption of 12%, whereas phenobarbital has a partition coefficient value of 23 and 30% colonic absorption is shown by it. Another parameter which influences the dissolution of the drug is the viscosity. In order to improve the physical characteristics of a dosage form like solutions and suspensions, viscosity enhancers are added which make the dosage form relatively viscous. Generally, the viscosity of a liquid dosage form may affect the drug absorption. In case of suspension, increasing the viscosity of a dosage form will lower the dissolution rate. Addition of methyl cellulose to solution results in decrease in the absorption of drug. Increase in viscosity leads to the reduction in diffusion rate of molecules in the lumen of the gastrointestinal tract. The gastric emptying rate is inversely proportional to the viscosity. Both effects can lead to reduction in the rate of drug absorption from the gastrointestinal tract, that is GIT. Employing colloidal diluents which become gelatinous upon contact with the aqueous biological fluid is highly undesirable in solid dosage forms. The more viscosity that would be produced immediately around the capsule or the tablet would limit the diffusion of gastrointestinal fluids to the drug to achieve dissolution and will further limit the dissolution of the dissolved drug to the absorption surface. This mechanism is utilized in several sustained release dosage forms. However, it is disadvantageous for drugs that are intended to be rapidly available. Now let us understand about the bulk solubility and the solubility in the different layers. The solubility of weak base or acid depends on the pH. Thus, there is variability in the rate of dissolution of such type of drugs in different areas of gastrointestinal tract. For weak acids, the saturation solubility, which is denoted as Cs, is given by the equation Cs is equal to Ha plus A, where Ha is the solubility of the unionized form of the drug and A is the solubility of the ionized form of the drug. When the solubility of the undissociated acid is C0, then Cs could be expressed in terms of C0 and dissociation constant Ka. Then the equation becomes Cs is equal to C0 plus Ka C0 upon the hydrogen ion concentration. This equation represents the solubility of weak acids, and the following equation represents the solubility of weak base, where Cs is equal to C0 plus C0 into hydrogen ion concentration divided by the dissociation constant Ka. When the expression is modified into the Noise-Whitney equation, the following equation is obtained for a weak acid as shown below, and for the weak base as depicted by the equation, dC by dt is equal to ds upon h into C0 1 plus h plus upon dissociation rate constant. Both the above equations demonstrate that the rate of dissolution of weak acids increases with increase in pH. On the other hand, the rate of dissolution of weak bases decrease with increase in the pH. Therefore, the rate of dissolution of weak bases would be optimal in gastric fluids, and the rate of dissolution of weak acids would be minimum in the acidic conditions of the stomach. These above mentioned equations are able to determine the rate of dissolution of weak acids in the small intestine and the dissolution of weak bases in the stomach. Under such environment, the hydrogen ion concentration of the bulk, which is represented by H plus, is not equal to the hydrogen ion concentration of the diffusion layer. For weak acids, the hydrogen ion concentration in diffusion layer is greater than the hydrogen ion concentration in bulk, and the reverse is applicable for the weak bases. The hydrogen ion concentration of the diffusion layer can be evaluated by using pH of appropriate aqueous buffer media saturated with drug. The next theory is the Danckwert's theory. Danckwerts does not consider the existence of diffusion layer. As you can see from the diagram, he suggested that in the dissolution media, there is turbulence at the solid-liquid interface, because of which the agitated media consisting of solvent packets reach the solid-liquid interface in a random manner because of the eddy current. The solvent packets absorb the solute by diffusion and carry them to the bulk of the solution. The solute-loaded packets are continuously replaced with new packets of fresh solvents. Since the packet of solvents are exposed to new solid surface each time, the theory is also known as the surface renewal theory. The Danckwerts theory is explained by the equation as shown below, where V is the volume of dissolution media, dC by dt is the dissolution rate, A is the surface area of the drug, and Cs minus Cb is the concentration gradient, and gamma is the rate of surface renewal, also referred as interfacial tension. The third theory is the interfacial barrier theory. According to this theory, an intermediate concentration can exist at the solid-liquid interface as a consequence of solvation process. In case of a crystal dissolution, an interfacial barrier exists for every phase of the crystal. This theory is clarified by the given equation, where Ki is the effective interfacial transport constant and G is the rate of dissolution per unit area. The second physicochemical factor affecting the absorption of the drug are the polymorphic crystals, hydrates, or the solvates. When a substance is present in more than one crystalline form, the forms are called as polymorphs and the phenomena is called as polymorphism. The drug ability to exist in different crystal forms may change the drug's solubility. Moreover, the stability of each form is very important, as polymorphs may get converted from one form to another. Polymorphs exhibit similar chemical structure but different physical characteristics. Physical properties include hardness, density, solubility, and compression properties. Many polymorphic crystals have poor water solubility in comparison to the amorphous form, leading a drug product to be poorly absorbed. For example, chloramphenicol has different forms of crystal. In case where it is administered orally as a suspension, the amount of the drug present in a body depend on the percent of beta polymorph in suspension, because beta form is highly soluble and well absorbed. There are two types of polymorphs. Enantiotropic polymorphs, it is the form which reversibly changes into another form due to changing conditions like pressure or temperature, and the example is sulfur. The next is the monotropic polymorphs. It is the form which is unstable. Example, glyceryl stearates. On the basis of stability, one of the various polymorphic form is physically more stable than the others. This type of polymorph represents the lowest energy state. They exhibit more melting point. Moreover, they show less solubility in water. The rest of the polymorphs are referred as metastable forms. These forms show a higher energy state and exhibit lower melting points and more solubility in the aqueous medium. Depending upon the internal structure of a compound, they can be categorized into crystalline and non-crystalline form, also known as the amorphous form. Crystalline form of a drug indicates that it has internal crystal structure, whereas amorphous form of a drug exhibits no internal crystal structure. Amorphous form of a drug shows more aqueous solubility in comparison to the crystalline form. This is due to the fact that the energy needed to disrupt a molecular lattice is higher for a crystalline form than that needed for the amorphous solid. For instance, the non-crystalline form of aceclofenac is more soluble in comparison to its crystalline form. A polymorph or a molecular adduct are the crystalline form of a drug. The stoichiometric kind of adducts, where solvent molecules are incorporated into the lattice of a solid crystal, are known as solvates. The incorporated solvent is referred as solvent of crystallization. When water is a solvent, then solvate is known as hydrate. Usually, the anhydrous form of a drug exhibit more solubility in water in comparison to the hydrates. The reason for this is that the hydrates are already in interaction with the aqueous phase and thus has low energy for crystal breakup in comparison to the anhydrates for further interaction with water. For example, the anhydrous form of ampicillin and theophylline exhibit more aqueous solubility, dissolve at a higher rate and show better bioavailability in comparison to the monohydrate and trihydrate forms respectively. The next factor is the chirality of a drug. Various drugs are chiral compounds. The enantiomers, that is the R and the S forms, or the D and the L isomers of the chiral compounds differ in pharmacological activity, biotransformation, dose, etc. Single enantiomer drug could be superior to its racemate, as the other enantiomers may cause side effects, like dextrodopa is highly toxic as compared to levodopa. The following table shows the advantages of a single enantiomers. The next factor is the salt form of the drug. Most of the drugs are available either as weak acids or weak bases. Formulation of a salt form of a drug is the strategy to increase the solubility and dissolution rate. If we see at the table, we will find that for the weakly acidic drugs, the sodium and potassium salts of barbiturates and sulfonamides have been prepared, and for the weakly basic drugs, hydrochlorides or sulfate salts of various alkaloidal drugs are available. Another example is avitriptan. Hydrochloride shows aqueous solubility of 3.4 mg per ml, whereas methane sulfonic acid salt of the drug exhibit 16.3 mg per ml solubility. Moreover, when it is prepared as succinic acid salt, it showed a solubility of 16.1 mg per ml. The reason for improved solubility of salts of weak acid is that the drug precipitates as fine particles as shown in figure. When a soluble ionic form of a drug diffuses from the diffusion layer into the bulk of the solution, which has a low pH, it is converted into its free acid form, exhibiting low water solubility at the low pH of the bulk solution. As a result, this free acidic form of the drug is precipitated in the form of fine particles. This leads to enhanced surface area of the drug particle, which causes faster dissolution and absorption than the drug administered in just the acidic form. Let us understand about the drug dissociation constant known as the pKa and the pH partition hypothesis. According to the pH partition theory, the drugs exhibiting molecular weight more than 100, their transport across the biomembrane is by passive diffusion. The absorption is controlled by the following parameters, which are the pH at the absorption site, the lipid solubility of the unionized drug, and the pKa of the drug. The amount of the unionized drug is a function of the drug pKa. Moreover, it is also the function of media pH. If we look at this, lower the pKa of the acidic drug, stronger the acid, higher will be the percentage of ionized form at a particular pH, and higher the pKa of the basic drug, stronger will be the base. Henderson-Hasselbalch equation is useful to calculate the content of the ionized form of the drug in the fluid at a particular pH. For the weak acids, pH is equal to pKa plus log ionized drug content upon the unionized drug content, and for the weak bases, pH is equal to pKa plus log unionized drug content upon the ionized drug content. The effect of the drug pKa and the GI pH on the absorption of the drug has been discussed in table. If we see, for the very weakly acidic drugs which have a pKa more than 8, like ethosuximide, phenytoin and various barbiturates, at all the pH values they are unionized and they show rapid absorption. For moderately weak acidic drugs having a pKa between 2.5 to 7.5, like ibuprofen and aspirin, they are well absorbed from the acidic conditions of the stomach because they exist in the unionized form there. For the strongly acidic drugs which have a pKa less than 2.5, like cromolyn sodium, they are ionized at all the pH values and thus they show poor absorption. For very weak basic drugs having a pKa less than 5, like theophylline, diazepam, oxypam, nitrazepam and caffeine, they exist as unionized at all the pH values and hence show rapid absorption, which is pH independent. For moderately basic drugs having a pKa between 5 to 11, like chloroquine, amitriptyline, imipramine, these drugs are affected by the variation in the pH and thus their absorption is pH dependent. They are unionized at pH of intestine and hence better absorbed from there. And strongly basic drugs having a pKa more than 11, such drugs are ionized at all pH range of the GI tract and thus they are poorly absorbed. Drug stability in gastrointestinal tract. A drug should be stable in its dosage form for its better physiological availability. In case if the drug gets destabilized in its solid or liquid dosage form before its administration, it causes less drug dose per unit dosage and consequently leads to lowered bioavailability. Not only the drug stability in the dosage form is important, but also the stability of the drug in the gastrointestinal fluids is also important. The wide range of pH and activity of enzymes in the GI tract causes degradation of drug. Hydrolysis is a major pathway that occurs in acidic gastric fluids and causes degradation of drug to inactive moieties. Erythromycin, which is an antibiotic, has a pH dependent stability profile. In gastric fluid, this drug degrades rapidly, but in the alkaline or neutral pH, the drug is relatively stable. Due to this reason, erythromycin tablets are available as enteric coated in order to prevent acidic degradation of the drug. The hydrolysis and inactivation of penicillin G in the acidic environment of the stomach also provides a good illustration. Surface activity. In almost every dosage form, like the solids, semi-solids and liquids, surfactants are utilized for various reasons. Surfactants influence the dissolution rate and thus the absorption. Reducing the surface tension leads to enhancement in the rate of dissolution by increasing the drug solubility. The decrease in surface tension also tends to enhance the free molecule diffusion in the medium, increasing the contact between the surface of absorption and free drug. Surfactants also enhance the membrane permeability, permitting more absorption. The surface active characteristics of gastrointestinal contents primarily involve the various fluids like the intestinal juice and gastric juice and can be the main factors in the dissolution of various lipophilic drugs. The key important component of GI fluids is the more content of bile salts present well above their critical micelle concentration. The micelles of bile salt solubilizes polycyclic aromatic hydrocarbons, cholesterol, progesterone and hence enhance their absorption. Complexation. A molecular complex comprises of constituents that are held together by weak forces like the hydrogen bonds. The properties of drug complexes like molecular size, solubility and partition coefficient can vary from those of the drug itself, leading to differences in the bioavailability. Complexation generally enhances the solubility of poorly water-soluble drug if the complex itself is soluble in the aqueous medium. The drugs which are usually adequately absorbed, the formation of complex may lead to slowing down of the absorption process, but the amount of the drug absorbed may not change. Various examples exist where rate of absorption has been enhanced as a result of complexation. Adsorption. Surface adsorption influences the bioavailability of drugs. Adsorption involves the removal of the molecule from the solution and their transfer to the surface of an active solid substance like charcoal, alumina or clay. Activated charcoal has long been utilized to reduce the bioavailability of drugs because of the large surface area it presents for adsorption by physical interaction through the network of bonds carbon structure. In same manner, materials like pectin and attapulgite reduces the adsorption of drugs like promazine. The forces of adsorption are for the most part weak physical interactions like the van der Waals forces, but often a chemical interaction is also possible and this is known as chemisorption. Now students, let us summarize this module. Dissolution is defined as the process of solubilizing a solid substance in a solvent. There are three theories related to the dissolution of drug, that is the diffusion layer theory, DANCKWERT'S theory and interfacial barrier theory. Crystalline form of a drug indicates that it has internal crystal structure, whereas amorphous form of a drug exhibit no internal crystal structure. Amorphous form of the drug has more aqueous solubility in comparison to the crystalline form. Anhydrous form of a drug exhibit more solubility in water in comparison to its hydrates. There is enhancement of solubility of salts of weak acids in the biological fluids due to drug precipitation as fine particles. pH partition theory states that drugs exhibiting molecular weight more than 100, then they are transported across the biological membrane by passive diffusion. Henderson-Hasselbalch equation is useful to calculate the content of ionized form of the drug in a fluid at a particular pH. Thank you.